Hello everyone, welcome to the latest Novogene webinar, Next Generation Sequencing, a Beginner's Guide. I'm John Cafferty, Marketing Manager here at Novogene Europe. This is the third in our lockdown webinar series. We'll be sending you details of the rest of the programme after the webinar. A bit of housekeeping. The webinar will last 1 hour with a 10-minute question and answer session at the end. Please submit your questions via the question and answer chat functionality you will see at the bottom of your screen. We'll answer as many questions as we can, but if we can't answer your questions live, we will get back to you by email after the webinar. I would like to introduce your host for today, Courtney Camboris. Courtney completed a PhD at the University of Maryland, followed by a postdoctoral fellowship at the Johns Hopkins School of Medicine. Her research focus was on transposon mutagenesis using next-generation sequencing. Since then, Courtney has worked as an account manager for various companies in the United States, the Netherlands, and France. She joined Novogene in February and is a regional sales manager for France. Courtney, over to you.
Thank you, John. And thank you for joining us today for our webinar. As John said, I'm the regional sales manager for Novogene in France. Before we dive into our webinar, I'd like to give you a short introduction to our company. Novogene was founded in 2011 by one of the world's leading experts in genomics and bioinformatics. Solutions. And high-performance computing. Novogene has one of the highest sequencing capacities in the world. Our cumulative output of all of our instruments is more than 280,000 human whole genomes per year. This is not only supported by our instruments in our laboratories, but also by our supercomputing data centers. Now, Novogene has a global presence. So it was founded in Beijing and our headquarters remains there, but we have subsidiaries across the world. So for the American customers, we have a lab at UC Davis in California. And for our European customers, we have a lab in the United Kingdom in Cambridge. And we're also starting a subsidiary in the Netherlands to even better serve our European market. So this is an overview of the research services we offer at Novogene. As you can see, we offer a very large variety of services. So if you're working on the human genome and you're interested in whole genome sequencing, whole exome sequencing, or target capture sequencing, we have the ability to do that. If you're focused more on animal and plants, we can do the de novo genome sequencing, whole genome sequencing, or reduced representation sequencing. If you're working on bacteria, you're one of our microbial customers, no problem. We have de novo sequencing there, we have resequencing, we have amplicon sequencing, as well as metagenomic sequencing. If your lab's focused more on gene expression, so if you're looking at the transcriptome, so if you're looking at RNA, we can do mRNA sequencing, non-coding, single-cell RNA sequencing, and isosync. If you're focused more on methylation or protein nucleic acid interactions, we have a variety of tools available for epigenetics research, including whole genome bisulfate sequencing, CHIP, and RIP sequencing. Now, if you're working on something that you don't see listed here, please let us know, because we can make a feasibility study for you and let you know if it's something we can work together on. So as I mentioned, we are a global company. However, we are also very well positioned locally. We have over 25 local sales representatives across Europe who combined speak more than 15 languages. So together we can help you achieve your research goals. Here's our lovely sales force. So we have representatives in DACH, in the Nordics, in Benelux, the UK and Ireland, in Southern Europe, which includes Spain, France, and Italy, as well as out in Eastern Europe. Easiest way to contact us is via email at europe@novogene.com. And we'll go ahead and distribute that to the proper representative. So now I know we've all been waiting to get into the webinar, so I'm going to stop here. Introduce our speaker, Zimin Zhuang. So Zimin received his bachelor's and master's degree from the University of Adelaide in Australia, researching the epigenetic effect of maternal high-fat diet. Prior to joining Novogene, he spent 2 years doing NGS R&D research on cancer biomarkers from cell-free DNA. After joining Novogene in 2018, he continued to develop his NGS expertise, starting out in technical support and product management. In August 2019, he was promoted to product manager supervisor and now heads up the Novogene Europe product department. Over to you, Zimin.
Thank you, Courtney. Okay, good morning, everyone. I'm really glad to talk to everyone at our Novogene webinar series. And my name is Zimin, the product manager of Novogene Europe. And I'm going to talk to you today about some beginner concept of next generation sequencing. The outline of our today's talk will cover a bit about the history of sequencing technologies. Then we will focus on the principle of NGS, major in the Illumina platform, which most NGS is being performed on today. At the end of today's talk, we will spend some time to discover the application of next generation sequencing and to see how Novogene can help you to achieve your research target. This timeline shows some key stage of development of DNA sequencing. In early 1950s, DNA double helix structure and the center dogma had been approved. 20 years later, the first DNA sequencing concept based on the chain termination method, or called Sanger sequencing, has been developed. This method even came earlier than the PCR reaction. In the next 20 years, this method had been widely applied to research area with the invention of the automatic sequencing machine. People usually call this stage as first generation sequencing. In 2006, companies such as Illumina developed a new sequencing method based on the sequencing by synthesis. This is also a milestone of the application of second generation sequencing. So today we will spend most of our time to discuss the Illumina technology. Soon after the development of NGS, companies such as Nanopore and Microbial announced their long-read sequencing method, which are also known as the third generation or the next generation sequencing. So let's have a quick look of different sequencing methods at different timeline. In 1970s, the DNA sequencing became a practical tool with the invention of the chain termination method by Fred Sanger and his colleague. This method adds a different fluorescence dye when the DNA synthesis terminates at a random length. At the beginning of this method, it's required a manually check of the gel and to interpret the sequencing results based on the electrophoresis. Later on, this method is packed into a commercial level with the help of invention of the automatic sequencer, such as this ABI 3730. This sequencer remains a dominant method for the DNA sequencing all the way through the Human Genome project. The benefits of Sanger sequence is the high accuracy, but the disadvantages is the low throughput and the costs are quite high. Each machine can only run up to 96 different DNA molecules with around 1,000 base pair. If to sequence a single human genome on one machine, this will require more than 100 years. So for the Human Genome project, it actually recruits hundreds of sequencers worldwide to complete this project. So the story of Human Genome project is really exciting, but scientists at that time can only sequence from only a few people. They still don't understand most of the genome and of course, most of other species. So another amazing story is what happens to the DNA sequencing after the Human Genome Project. And that can be simply explained. It shows the cost to sequence one human genome. This dropped significantly from 100 million USD at 2001 to around only 1,000 USD at 2008. So what happens in these two decades? If you still remember the timeline I showed in the first slides, so in 2007, a new sequencing method started to be used in the scientific lab around the world. That is the next generation sequencing or the second generation sequencing. This method is sometimes called a massive parallel sequencing because this method allows to sequence millions and billions molecules of DNA at the same time. So later on, we will explain in detail to see how the sequencer is able to achieve this. So the improvements are not only the price, but also reduce the labor and increase the speed of sequencing. Over time, the next generation sequencer has become the premier tool to study the nucleic acids such as DNA and RNA. These tables here shows how significant next generation sequencing improved the performance. So for example, with the new Novogene 6000 machine, we can actually sequence the human genome for less than 1,000 USD with only about 40 hours run on the machine time. One of the shortage of NGS and Sanger sequence is that the read lengths are quite short. So for a typical Illumina sequencer, the fragment generated is around 500 base pair, while the Sanger can reach around 1,000 base pair. For some methods, such as genome de novo assembly, there is a need to sequence very long lengths. To achieve this, the next generation or the third generation sequencing had been invented. This technology can sequence up to 20 kb of the DNA sequence. And we will not spend too much time talking about third generation sequencing today. And as I mentioned, we will only focus on the principle of the Illumina sequencing. Illumina has developed a series of sequencers. Some are the benchtop sequencer. This machine can be easily installed and run in the individual labs. However, the running costs are quite high for an individual lab, and the data throughput are relatively low. This is why they also have the production scale level, which are used for companies such like us. So, for example, Novogene 6000 is also our main platform at Novogene. The sequencing of this Illumina platform has to happen in these flow cells. These flow cells are microscope slides with channel on the inside surface. So this is the workflow for a typical Illumina sequencing. The typical Illumina sequencing workflow is composed of 4 basic steps. The first one is called library preparation. So library preparation is to prepare the DNA and to make it able to run on the float cell. Next step is cluster generation. Cluster generation is an amplification reaction that occurs on the surface of the float cell. Then the reads are be able to sequence and the data can be used for analysis. We will go to see the details step by step. To begin with, you cannot just put DNA directly into those flow cells and get the sequence out. You actually have to prepare your samples. Basically, a DNA of your studied species are extracted and shared into pieces by either sonification or the restriction enzyme digestion. cDNA reverse transcript from RNA are also available. Then the fragments is repaired and the adenine is ligated to the end of the sequence so that the adapter can be added to these fragments after a limit number of the PCR reaction. This whole step is called library preparation, and now the libraries are ready to be sequenced. Let's have a close look of the library structure. The DNA or cDNA of interest is shown in the middle as the black color. During the PCR reaction of adding the adapter, the additional oligos are also introduced. These will include sequencing primer binding site, which shown in yellow color, indices shown in red, and regions that complementary bind to the float cell oligo, which were shown in purple. Here I also introduce some other basic concepts about the sequencing. A single end read is when only read one primer binding to the primer binding site and sequence in one direction. A PE read, which means the pair end read, is when both ends is being sequenced. A single index library is that the library has one index at one end, while a dual index is a library that has two indexes at the both ends. What are the purpose of these indices? As I talked before, the NGS enabled to sequence a massive number of molecular with very, very high throughputs at the same time. Therefore, it is possible and cost-effective to sequence more than one sample at the same run. In order to identify the reads according to its samples, multiplexing is needed. In order to achieve this, an individual 6 or 8 base pair index sequence are added to each sample, and they work like a barcode. So they can be differentiated during the further data analysis. Once library is prepared, the next step is called clustering. Clustering is a process where each library molecule is isothermally amplified. So as I mentioned, the flow cell is a black slide with lanes. Each lane is also coated with two different types of oligos. So this is the flow cell's surface, and each flow cells, they got quite a lot of oligos on the surface. The double-strand library structure will be denatured into single strand and flow into the flow cell. Hybridization is then occurred. So the two types of the oligo on the surface is complementary bind to the adapted region of one of the fragment strands. A DNA polymerase synthesizes the complementary strands of the original hybridized fragment to form a double-strand moleculus. This double-strand moleculus is then denatured, and the original strand is washed away because the original strand doesn't attach to the flow cell directly. So only the new synthesis strand will be remained. The strands are then clonally amplified multiple times through a process called this bridge amplification. In this process, the remained newly synthesis strands are flipped over, and the adapted region at the other side is hybridized to the other type of oligo on the flow cell. Polymerase synthesis the complementary strand, forming a double-strand bridge. This bridge is then denatured, resulting in two single-strand copies of the molecules that are attached to the flow cell. This bridge amplification is then repeated over and over through numbers of cycles of PCR reaction. All these steps occur simultaneously for millions of clusters. So this finally resulting in a clonal amplification of all the fragments in one lane. After the cycles of bridge amplification, this bridge is then denatured, leaving millions of single-strand DNA on the flow cell. After the amplification of the reverse strand, these reverse strands are cleaved off and washed off, leaving only the forward strand. So as the graph shows. The 3' ends of the forward strands are blocked. This blocking is to prevent unwanted binding again between the 3' ends and the cleaved oligos. So now the clustering has been complete, and the fragment structure is now able to be sequenced. The key method Illumina based is called sequencing by synthesis, SBS. So sequencing began with the read one. Read one primer binds to the read one sequencing primer binding site of the adapter, and the read one will be generated along the DNA synthesis. With each cycle of DNA synthesis, different fluorescence tagged nucleotides A, T, C, or G will complete to each other for additional on of this growing chain. Only the one that are matched to the templates will be added. After the addition of each nucleotide, the cluster will be excited by a light source. So the light source comes in, flash onto the float cells, and the signal is reflected back to the machine. This is why this method is called sequencing by synthesis, because each cycle, this will generate a signal. The number of cycles of this extending is pre-set up before the sequencing was run. Mostly 150 base pair will be extended. The machine will automatically record all these signals, and these signals will finally transmit to a base call. For a given cluster, all those identical strands are read simultaneously, and millions of clusters are sequenced in a massively parallel process. After completion of the first read, the synthesized read product is washed away. The next step is to read the index. The index 1 read primer is then introduced, so which is the I7 index primer. It's bound and hybridized to the index sequencing binding region of the adapter. The index read is generated similar to read 1 and recorded by the sequencer as well. After the completion of the index read, the read product is washed off, and the 3' ends of these templates are deprotected. If the sequence is set up as pair-end read sequencing, the reads are then flipped over and bound to the second oligo of the float cell. Index 2 is read in the same manner as index 1, and the index 2 will be also recorded. Index reads are then washed off, and the polymerase then extended to the second float cell oligo to form a double-strand bridge again. This double-strand bridge DNA is then linearized, and the 3' ends are blocked again. The original forward strands are washed off and leave only the reverse strand this time. So finally, the read 2 begins with the read 2 sequencing primer. As same as the read 1, the read length will be extended until the desired read length is achieved. So the read 2 product is then washed away. This entire process generates millions of reads representing all the fragments. Now the sequencing are completed. Sequences from the pool samples libraries are demultiplexed based on the unique indices, as I mentioned, during library preparation. Those sequences are now ready to be used for data analysis. For Illumina platform, they also offer a series of flow cells and the reagent to meet different inquiries regarding of different budget, different throughput, and different output. The common kits that large sequencing providers use are S4 reagent and SP reagent. So the ones circled in red box are the key reagent that Novogene are using. This can meet the needs for most of the common sequencing strategies, such as P150. This means a pair end, which each end will extend for 100 base pair, a pair end 250 base pair, and pair end 50 and SE50. The quality of the sequencing is determined by the Q-score. A Q30 is considered to be a benchmark for quality in next-generation sequencing. A Q30 reflects that the base call is 99.9%. For Illumina officially guarantee, they guarantee a Q30 over 75%, which means 75% of the reads has more than 99% accuracy rate. So for most of the service we provide at Novogene, we can guarantee a Q30 over 80% if the sample pass our quality control. So this is actually surpass Illumina's guarantee. The table shows on the bottom right is showing the comparison between different Illumina float cell. Using this NovaSeq S4 float cell as an example, the average read length is P150. Each run can load 2 float cells, and each float cell has 4 lanes. The total throughput for one run is 6 terabase pair, which equals to 20 billion reads. The runtime on the machine itself only requires 40 hours. The sequencing results will be stored as a FASTQ format, which can then be used for further analysis. The first step is called data QC. In this step, some reads with the low quality will be filtered out, such as reads with low quality or the reads that has uncertain base reads. And also they filter the reads that contain some contamination, such as adapter contamination. Next step, the read will be aligned and mapped back to the reference. This step is called data mapping. After that, different types of analysis can be applied according to different library type and research need, such as mutation calling for DNA sequence and quantification analysis for the RNA sequencing. Okay, so as you see, that next generation has a really completed system, and how this next generation sequencing will help with the research. NGS is widely used in many molecular biology studies. So here is the center dogma for genetics. The DNA carries the genetics information, which will transcribe to RNA. RNA expressed differently to make functional product, which is the protein. Protein is the basic block of all the cells of our body. In this process, NGS can be applied to study the DNA of different species by genome sequencing, and also differential gene expression can be studied by various transcriptome sequencing study. And epigenetics differ from genetics by the fact that epigenetics can alter the gene expression without changing the DNA sequencing. This includes mechanisms such as DNA methylation, a histone modification, and a microRNA binding interaction. All of this can also studied with the help of NGS. Okay, so as a world-leading NGS service provider, how Novogene can help you to achieve your research target? We offer a wide range of service. If your lab have the ability and want to make the library by yourself, what we can offer is a sequencing-only service. We will evaluate your personal libraries case by case to check if it's compatible with our Illumina platform. The recommended size for the library is around 300 to 500 BP. For the data throughput, no matter you only require 1 gigabase or you require a whole flow cell, it is all capable for us. After we receive the library, we will perform a strict QC to check the quality and the concentration of your library. If everything works fine, we will run the sequence and the raw data will send back to you for analysis. We know that the turnaround time is very important for most of the studies. So we also offer the express service and the different circumstances. If you are interested in this express service, you can check with your local account manager. A good quality of library will ensure a good sequencing result. So we also recommend you to perform a pre-QC before sending us the library. Here is some Agilent 2100 results of the libraries, and I would like to share some experience with you to see how we can get good quality library. The one on the left side reflects a very good quality library. As you can see, there is only one peak according to the correct size, and the peak range is quite narrow. There is no smaller fragments at the beginning, and there is also no large fragments at the end, so which means there is no DNA fragment contamination. The one on the top right had some small fragments at 60 BP and also around 130 base pair. So this indicates adapter and an adapter dimer contamination. Since the adapter at one end is around 60 base pair, and if you combine the adapter dimer, that will around 120. If this occurs, a purification can be applied to remove the small fragments post-library preparation. But if you want to avoid this during library preparation, what you can do is just simply adding less adapter into the system. The one on the right bottom has some tailing issue. So this tailing issue will significantly affect the total output of the sequencing. To solve this issue, a set selection step can be added during the library preparation. If your lab does not have much experience with sample preparation and also library preparation, what we can offer is also a package service. So for this, we can help with sample preparation. We also run a strict quality control to make sure all the samples pass the quality and can be used for library construction. We will perform the library construction, sequencing, and we also offer a comprehensive analysis for different studies. As I mentioned, a good quality DNA or RNA will ensure the quality of the sequencing result. Therefore, we will run the QC before every library preparation step. If your research interest is in the human genome study, for example, if you want to study a genetic variant between a population, or look for some disease-associated mutation, a whole genome sequencing can be used to scan the whole genome level from different species. If the DNA of your interest is only in the protein coding region exon, and you want to sequence the exon in a more deep depth, a whole exome sequencing can also be selected. This method requires a capture of the exome sequencing, which can be selected by the beads. The whole exome sequencing is also an ideal method for low-frequent mutation. If only a list of the gene you want to sequence, there is also a service called target region sequencing. So for this method, we can help you to design the experiments in different cases. Here is a case study I would like to share with you. So Novogene was participates in the large-scale WGS, in which the whole genome from more than 4,000 Singaporeans was sequenced. And this studies was used to identify genetic diversity between 3 different acidity groups. 52 millions of the novel SNP, so this SNP is the single nucleotide polymorphisms, and the indel is the insertion or deletion from the genome has been founded. This also provides an insight into the population structure and the evolutionary history of the Asia. For plant and animal genome study, a genome resequencing aims to resequence the individual whole genome if the genome sequence is already available. The major application to this is to detect the genetics variants, including rearrangement, repetitive sequence, and the fusion genes. Novogene is also provide leading service for the de novo sequencing to sequence a novel genome. Basically, to sequence a novel genome, a combination between the short reads and the long reads is the ideal way to maximize the coverage and improve the sample performance. So this requires a combination of these two methods. We will not cover too much detail about de novo service today. And as John mentioned, we will arrange another webinar talking about different service lately. So if you are interested in this, please also follow our webinar schedule. Here is some genome assembled studies Novogene that has helped research to published. As you can see, we have many experience with this type of studies and also the experience with many interesting species. NGS has also played an important role in microbial genome study. So for a given isolated culture, if the individual bacterial or fungal genome DNA can be isolated, a de novo or resequencing can also be applied based on whether the reference genome is existed. If the studies is focused on the macro environment, such as human intestine, the feces, and also from the wastewater or soil samples, the total DNA can be isolated from the macro environment. A metagenomics can be used to study the microbial mixture. So if a specific region is amplified and sequenced, this is called an amplicon-based metagenomics. For example, amplification of 18S and ATS can be used to study the fungal and algal diversity. A 16S can be used to study the bacterial diversity. If the whole genome is being sequenced for the total DNA isolated from the macro environment, this is called a shotgun metagenomics. This is not only show the microbial diversity, but also study the gene function of different species. This is an example of an amplicon-based sequencing study. This study is trying to figure out whether the translocation of the human gut microbiome after getting a type of cardiovascular disease, STEMI, will induce other side effect. So with the help of sequence the microbiome from the blood, results show a richness of the gut-dependent microbiome after getting this disease. A heat map also show the general difference between different groups and their phylogenetic relations. The top 40 most different genera across the group can also be identified. So with the help with all this data, other different analyses can be used, such as to study the original of these different microbiome composition, and this can be used to prevent further negative side effect. The transcriptome and regulation service is still the research hot point, and we can offer a variety of the service for transcriptome and regulation service. For the transcriptome study, if the reference gene is available for an eukaryotic species, we can offer mRNA-seq with either NGS or the Microbial platform. For the non-coding RNA, we can offer long non-coding RNA, circular RNA, and small RNA with the NGS platform. The whole transcriptome can be used to study the correlation between coding and non-coding RNA. For prokaryotics, what we can offer is RNA-seq as well. If the reference gene are not available, that's still okay. We also have the analysis pipeline for the de novo transcriptome study. For epigenetic study, we can offer a ChIP-seq study to study the DNA and protein interaction, and the RIP-seq to study the RNA and protein interaction. To study the DNA methylation, we can offer the golden method, which is the whole genome bisulfite sequencing. Methods such as reduced representative bisulfite sequencing is also available. Here is an example of the long non-coding RNA study. So with this strategy, we can sequence both information for the coding and for the non-coding RNA. The differential gene expression can also be studied. This heat map is used to find genes with similar expression patterns and the various experimental conditions. By clustering genes with similar expression pattern, it can also be possible to predict the unknown function of the previously characterized gene or the gene functions. This study has also performed an integrative analysis of long non-coding RNA and RNA, because the most important function of the long coding RNA is to regulate the gene expression. So the study can also able to achieve this. Overall, Novogene provides many different research services. If the services you are interested are not shown in this list, you can always contact with your local account manager. We can help to evaluate individual projects and to see if any sequencing-based technology can be incorporated to your studies. Here, we also summarize different sequencing strategy using different service. This is also based with our experience. If you have different inquiries, you can also discuss with our support team. So for example, to study WGS, the whole genome sequencing, WES, plant animal WGS, RNA-seq, long coding RNA, and all these other type of service, we recommend a PE100 with the NovaSeq 6000 S4 platform. At the end, I would also like to share this information with you that we also have the world-leading computing infrastructure and the server. So this provides us the capability to storage a huge amount of data and also give us the ability to analyze different sorts of analysis. So this is the end for today's talk, and thanks a lot for everyone's time. And I think now we can move to the question and answer part. Thank you.
Thank you very much, Zimin. That was excellent. So as Zimin said, we're going to move on to our question and answer part of the webinar today. I'm going to go ahead and read out the questions, and then Zimin will answer them to the best of his ability. If we don't answer your question live, we'll respond to you via email. So Anna has asked, what are your platforms for sequencing?
Okay, so the major platform that we were using are the NovaSeq 6000 platform. We also have the S4 flow cell and also have the SP flow cell. That's the major platform that we were using.
Great. Laura Bethel has asked, what is the longest read we can obtain from the Illumina platform?
So for the Illumina platform, the typical maximum read length we can get is 500 base pair. So this will require us to use the NovaSeq SP flow cells with the PE 250. So for this one, we can sequence 200 base pair at both ends. And in total, it will give up at 500 base pair.
Okay. And Leon Johnson asked, once the project is completed, how can I get the data?
Okay, so we have different methods to deliver the data to our client. If the data size is quite small, we will just transfer from the Amazon online network. And if the data size is large, we also can deliver the hard drives to our client. So we have different sorts of hard drive, different storage to check different, meet different inquiries. Yeah.
Perfect. Reed Clark has asked, how do I pull sequencing libraries? Can you pull them for me?
Okay, so for the individual library that you have made, we recommend you to run a qPCR reaction first to check the molarity of the libraries and pull them in the same molarity. If you cannot do the pooling, what you can do is send us the individual library. We will run a QC to the individual libraries and will help you to pull the libraries based on your need.
Okay. Brody Fecker has asked, do you accept PCR products instead of nucleic acid for RNA sequencing?
For the RNA sequencing, what we normally accept is the total RNA. And we do have some experience for some customer that they send us the complementary DNA after they perform the reverse transcription. But for the PCR product, it will need to be evaluated.
Okay.
We need to see what is exactly the purpose of the study.
Okay. Okay. Ray Woog has asked, how about the sequencing depth? What would you recommend for a de novo study?
So for the de novo study, the sequencing depth is actually dependent on how well you want to get the sample. And this is also evaluated based on the different projects and of course the different species. But for our experience, we normally combine an NGS together with the Microbial platform. And if this species that has not been studied before at all, we will use the NGS data to perform a genome survey. So we will check the complexity of this species. And based on these results, we can give a better suggestion on how much depth we need for the third generation sequencing. So, basically from our experience, most time we require 100 times for the NGS and around 80 times or 50 times for the Microbial. But this is actually dependent on case by case. So we cannot give a detailed number at this moment, but if you are interested, please contact with your local account manager. We will help you to make a plan.
Thank you. Miguel Ligon has asked, he has some samples with only a few picograms of DNA, and they'd like to know the bacterial structure. Can we sequence that little of volume? And what service should I send the order to? What kind of service should I use?
Okay. So for the DNA with extremely low amount, we do have some experience with the DNA amplification. So we will amplify the DNA first and to see whether the DNA is enough for us to continue with the library preparation. But to perform this, there will be a risk. So we will need to check how less the DNA you have and also to check the DNA quality. To study the viral structure, the bacterial structure, what we can do is to do a bacterial whole genome sequencing and compare to the reference genome to see if there is any structural variations. And if this is a bacteria that has not been studied before, we can also do a bacterial complete map or a bacterial drug map. But this amount is quite low, so maybe the bacterial drug map is the ideal choice.
Okay. I apologize in advance if I've mispronounced your name. Ayal Waghe has asked, in eukaryotes for mRNA sequencing, which one sequencing method is more adaptive, like NGS or PacBio?
So for the messenger RNA sequencing, the application between different platforms is just have the different need. So for NGS, the RNA will be fragmented and we won't sequence the full-length RNA. And for the PacBio, what we do is called an iso-seq. So the iso-seq can also be used to study the isoform and different other purpose. So and also to study the structure for the iso-seq. So based on your different research needs, you will choose different platform. if your concern is only on the differential gene expression, actually the NGS is enough for your research need. So it depends on the different needs.
Okay. Esther Ewaluwegmiga has said, there's some argument between the use of sequencing and genotyping. So what argument would you give supporting sequencing as a better option?
So for the next generation sequencing, normally people use the next generation sequencing to do the, for example, if it's a WGS study, they use NGS to do a whole genome scanner. So they sequence the whole genome level and to find some identical targets. Then all these findings will also need to be proofread by the methods such as Sanger or the qPCR. Because I like mentioned, the Sanger sequencing or actually the PCR, they are more accuracy. So the NGS can be used as a widely scanning, and then each genotype or the differential SMP need to be proved by other technologies.
Okay. Marissa Sinenovic has asked, in terms of personalized medicine, could you share what are possible advantages of Illumina short-read sequencing in detecting gene mutations that cause disease or rare mutations?
In case of the personal medicine, okay. So for this, I probably need to check with other specialties in the clinical area. So based on my knowledge, the NGS can, like I mentioned, the NGS is like a scanner to scan some, so to scan the SMP. And actually to study this type of studies, it is much more accuracy than the Microbial platform, and it has more depth.
Okay. Why don't we do a couple more questions, because I think we're running a little short on time. So for ChIP-seq, for broad chromatin marks, is there a sense to do sequencing more than single N50?
So for ChIP-seq, we do recommend 10 million reads to study the histone modification. And if you're interested in the transcriptome factor, we will recommend 20 million reads. So the sequence steps for the 10 million reads is sufficient for this purpose.
Okay. And she had a second question, was the best sequencing depth for the fruit fly. So do you think 10 million is sufficient for that?
Yeah, so what we can normally do is run a 10 million reads sequence to check whether they got their target's results. If the sequencing is not steps in deep enough, what we can do is also increase the sequencing to increase the sequencing depth. And they can also provide some extra materials. And also in that case, like if 10 million reads is enough, we can make a second library. And it also depends on the research needs, research purpose.
Okay, and then for the last question, do you perform variant calling from RNA-seq data, or do you suggest exome sequencing? Talking about coding region variants.
So for the exome sequencing and the mRNA sequencing, we do map the reference genomes. So for the WES studies, the benefit is its high depth. So we will sequence the exome region in a high depth. So the SMP call from the exome will be more accuracy. And for the RNA-seq, of course, we can also do the SNP calling. But if the mainly research purpose is just to detect the variants, we do recommend to use the WES. And another benefit for the RNA-seq is also to see what is the really expressed one. So for the WES, we just compare the DNA to the genome, but for the messenger RNA, we check the protein, the coding RNA, and that will show what is really expressed. So the purpose is different. And of course, the WES has more depth, and the RNA can be used to study the real expressed.
Okay, wonderful. I'm going to squeeze in one more short question, Zimin, sorry. Does Novogene provide a lincRNA mRNA modification detection service? For example, sequencing mRNA and showing the M6A modification location in the sequence?
So the M6A is widely used in the RNA methylation. And for this, what we can do, the M6A, the clients can do the enrichment by themselves. So it's like the RIP-seq. They will do the pull down, and we can accept samples after the pull downs and perform a RIP-seq. For the long non-coding RNA, what we normally provide is just a basic long coding RNA service.
Okay.
Perfect. And that's required total RNA.
Okay. Thank you very much, Zimin. I think we're going to stop doing the live question and answer now. We're going to leave the Zoom open for a little bit longer. So if you have any questions, please go ahead and ask it, and we'll answer it via email. I want to thank everyone for your time today. We hope you enjoyed the webinar, and we hope you found it useful. We will email you all a link to our webinar recording, as well as the slides, along with a very short survey. We'd really appreciate you taking the time to complete the survey. This survey helps to ensure that we make future webinars more relevant to you and the research goals you have. In the meantime, please stay in touch with us. You can follow us on Twitter and LinkedIn, or you can get in touch via our website at ennovogene.com, and we'll go ahead and forward you to your proper representative. We look forward to seeing you all at the next webinar. Thank you very much.
Thank you very much.